Good morning, Esther. I hope you all are fine with all your activities and enjoying at home with your preparations, with your family members, and doing nicely for the examination because very less days are there for the examination. So, in this present scenario, we'll proceed with our studies. As you know that we are discussing about the botany semester second paper number six, named as histology and anatomy and embryology. In that one, we are discussing about the embryology, and we finished different aspects related with the embryology, special reference with formation of male gamete, then formation of female gamete. Then we already finished about the pollination and then double fertilization process. Now, after this fertilization, after the double fertilization, what changes takes place in that particular embryo, in that particular ovule or inside the flower that we are supposed to study. In particular, we have to formation of embryo, then endosperm, then formation of seeds. In this way, the post-fertilization changes takes place. In this way, the first stage is formation of embryo, and that just after double fertilization it starts, and that we are supposed to discuss with a reference to this formation of or development of embryo. If you see that that pollen grain reaches on that particular stamens and stigma and then germinate and then fertilize, after that one they undergo certain resting periods. It required a particular resting period, as you know that it is given over here. In case of Astraceae family, it required 4 to 6 hours, while in Theobroma, Cocoa or Viscum, it required around 3 to 4 months this resting period. And after that one, actually, the division and revision process start. It means that whatever the zygote formation is there, that zygote undergoes division and revision process, and it is called as a embryogenesis. And that embryogenesis that results in the formation of embryo, and that initial embryo is called as a pro-embryo. This embryogenesis takes place in angiosperm in different way. There are different types that is mentioned in that one, and in different plant such type of division revision process continue, and that results in the formation of embryo. According to Maheshwari, mainly they are having two important types. The first one is crucifer type or onager type. Second one is astar type. According to your syllabus, you are supposed to study this first type, that is called as a crucifer type or onager type. In this, the basal cell which is present in that zygote, that plays the very important role, and that results in the formation of crucifer type of embryo. And the example is Anonaceae, Brassicaceae, Orangeaceae, Ranunculaceae, Scotocaceae. This Brassicaceae, initially it is called as a crucifery. That's why the name is considered as a crucifer type. Then another example on the basis of apical cell, then they are having solenite type, and these three types are there: caryophyllite type and chenopodal type. However, we are not supposed to study about this one. We are supposed to study this crucifer type of formation of dicot embryo, and that is our part of study, and that we are supposed to study in detail. This methodology is explained by Bhandari and Asnani in 1968, and after that one, Maheshwari and other scientists that has considered it as the ideal one. They have studied this one in crucifery family, and that's why it is called as a crucifer type of embryo. And they also studied this one on onager. That's why it is also called as a onager type of embryo. Actually, what's happened? Then the fertilization takes place inside the embryo cell, and that fertilization forms such type of zygote, as you know that very well. Now it is this zygote is a diploid one, because one haploid gamete fuses with this another haploid one, and that form such type of zygote. Then this such type of zygote undergoes division and redivision process. It goes mitotic division initially. क्या होता है उसमें? Then the single zygotes, nucleus divides and form two cells. And these cells are called as a apical cell. In short, it is called as a CA. And second one is a basal cell, it is called as a CB. In this way, apical cell and basal cell formation takes place. As you know that from the chalaza and food material comes inside, and this division and redivision process continue. This is the natural phenomenon. If the cell is there, then cell divide and redivide and form the many cells. Nucleus is also there, and nucleus, if nuclear cell is there, then again it divides and redivide and form the many cells. That we will study while we will start with the histology, special reference with the meristematic tissues. उसमें भी जो हमको ये सारी चीजें पढ़ना है. Then after that one will find that it again goes divide and redivide and form the median cells. This division redivision process continues in different stage. ये एक से दो, दो से चार, चार से 8 cell structure होता है. और 8 cell structure जब form होता है, so then again the apical cells as well as basal cell, ये apical cell और ये basal cell, again it divide and redivide and form number of cells. These are called as the initial cells. ये cells जो है तो divide और redivide होके they form such type of multiple structure, where on apical site a large quantity of this cellular structure formation takes place. It means that at the apical site a group of cells formation takes place, while towards the next site a particular number of cells are present that are 8 to 10 in numbers, and towards the initial cell site means toward the base side the cells divides and redivides horizontally as well as transversely. Then what's happened after that one? You will find that this division continue, and this division continue and result for the formation of such type of structure towards the apical side, means towards the micropylar end, and towards the basal side we will find that such type of structure develops. Later on what's happened? दिस ये जो अपाइकल सेल आपने यहाँ 1 2 में देखे थे, फिर 4 में देखे थे, फिर 8 में देखे थे, फिर मल्टीपल सेल स्ट्रक्चर होने के बाद वी विल फाइंड दैट सच टाइप ऑफ complex structure formation takes place, and this is considered as a apex part, where you will find that out toward the upper side the initial shoot apex structure development, where the dermatogen layer means the second layer is present, which divide and form different cotyledony layer structure. Towards the lower side again different cells are present, and that is called as a root apex structure and covered with root cap. In this way this complex cell division takes place. This is the stages of embryo formation, remember that. Again this division and redivision continue and form such type of structure where a large quantity of cells are present over here in this way, in this particular manner, and you will find that central notch start to develop. ये सेंट्रल नॉच जो है तो यहाँ पे बनना शुरू होता है. दिस सेंट्रल नॉच again continue in this way, in this particular form, in horizontal manner. उल्टा कर लिए तो ये बढ़ गया, और बढ़ने के बाद इस तरह का ये deep notch like structure form किया. Ultimately it results the formation of this two layer structure, and this two layer structure then contain a large quantity of cells, and in dicotyledon these two layers formation takes place, and it is called as a cotyledon. In case of monocotyledon only one layer formation takes place, but remaining part remain as it is, that is a pleume. Then toward the lower side this plumal formation takes place. Then colorizer, complete covering protective layer is there, collectile is there. This is the root apex structure which you find over here, and the same structure if you see, so you will find this type of embryo site. This is the wall, entire wall of embryo site. In this, inside this, such type of changes taking place, and these changes are resulting in such a way, in such a formation. Hence, This phenomena is most important, and that results in the formation of these two cotyledon. In case of dicotyledon, such type of embryo formation takes place where the endosperm start to germinate. इस तरह से ये embryo, this structure is called as a embryo. After formation of this one, then endosperm germination takes place. In this way, this embryo formation takes place, and this shows a particular division process which results in the formation of such type of structure. It is called as a crucifer type of embryo, and this is studied in the member of cruciferi. Name of the plant is a Ceratocephalus falcatus, and in that one you will find such type of formation takes place. So remember that one, how that zygote convert into embryo, dicotyledon embryo. That is, if you got the idea, it's very simple. Division, redivision process, formation of many cells. Then notch-like structure is there. That notch enlarges and form two cotyledons, and that's why it is present in dicotyledon. In monocotyledon, that not like structure continue but form only one layer, and that's why it is present in monocotyledon. So this process is called as a formation of embryo or post-fertilization changes after the formation uh, for the formation of embryo. And simply, this is embryogenesis. Simply, it is the formation of embryo. You are supposed to know this diagrammatic representation, although it's an objective type question examination. Otherwise, you are supposed to explain on the basis of this diagram which I am explaining you in that methodology. You are supposed to explain, but try to understand different stages: apical cell, basal cell, initial cells, cotyledons, then plumule, then radical. All these terms you are supposed to remember, and the same thing we'll discuss in detail about the endosperm. Okay, so you are supposed to be careful about it. You are supposed to prepare these notes. You are supposed to go through this one. All this process I'm also sharing with you in a video manner. Like yesterday's I'm shown you the double fertilization mechanism, very beautiful video. This one also I will share you with very beautiful video. Till then, take care of yourself, prepare yourself in nice manner, do hard work, try to understand this concept, and feels any doubt, any difficulty, ask me. Thank you. Bye bye.